For our next presenter, menjemput Profesor Emeritus, silakan. Ya, Prof. Prof Wari.
Sekejap, saya tengok susunan saya.
Maaf, Prof. Okey, saya jemput Puan Ku Ivi. It's on Salmonella, isn't it? Yes.
Good morning to everyone. First of all, my name is Ivi, and I'm a research officer from Veterinary Research Institute Penyelidikan Veterina. The location is at Ipoh. So this is my master project under the HLP, Tajan HLP, which with the title Characterization of Salmonella enterica, subspecies enterica, Siroa brachester, isolated from live chicken and raw chicken meat. So for the introduction, it's about the bacteria first. So this bacteria, Salmonella, is from the family Enterobacteriaceae. They are the gram-negative rod-shaped bacteria, and then they are the foodborne pathogen. And then this Salmonella mostly is classified into two clinically distinct groups, that is the typhoidal and the non-typhoidal Salmonella. So the Salmonella that I'm studying on, the Siroa brachester, is under this NTS group, non-typhoidal Salmonella. So next we look at the disease itself. So the Salmonella, they will cause salmonellosis in animal. So some of the animal, let's say in chickens, some they will come out with the clinical sign, but very few. Maybe they got the diarrhea, and then but most of the Salmonella harbors in the healthy chicken. They show no clinical sign at all. And then Salmonella in human, the disease is of public health concern because the clinical sign would be the self-limiting gastroenteritis, meaning after makan they will have the diarrhea and vomiting. And if those with the severe complication, much more serious one, they will end up with the infections such as pneumonia, fever, and some with the systemic infection until the blood level. They will, this term we term as the invasive NTS infection. So in my department, DVS, so DVS is the authority to control salmonellosis in animal chicken, in animal food products, and also the livestock animal. So our department carry out the National Salmonella Surveillance Program since the 1980s until now. So every year we are doing this. And then from the data in the 1990s, enteritidis is the most common Siroa that we identify in chicken, also in human salmonellosis. However, when we look at our data, we see that there is a shift in the prevalence, where brachester is coming up since the 2017. Here, this graph. So from there, we are concerned why suddenly there is a shift and then so many brachester isolated from our chicken. So that is where I start my literature search. When I Google about this Siroa brachester, there is not much information about this Siroa. We just know that it is placed in the group B alongside with other common Siroa such as typhimurium. And typhimurium actually is a penyakit wajib lapor in DBS. So from my literature search, I can see that this Siroa, the first reported outbreak in human was reported in 1952 in London, where it caused a death in infants and elderly patients in the hospital. And then later on, in 2001, there is reported cases of this Siroa brachester from the poultry and human cases, and they were found to be similar. And this paper, they suggest that maybe the isolate has the potential to become zoonotic. And then in our ASEAN region here, since 2017, there is more publication from Malaysia, China, and Singapore that also noticed that this is the dominating Seroa from the poultry and all along the processing plant. And until now also, this Seroa is the top 5 most common in our country. So the problem statement is stated here. The first one, Salmonella Brancaster, is the dominating Seroa, and currently there is limited further characterization study at the genomic level of the bacteria itself. And then our existing conventional serotyping is insufficient to understand the characteristic and the potential pathogenicity of the Seroa compared to molecular characterization technique. So the objective of my study is to investigate the virulence profile of the Seroa Brancaster. And then second is to determine the antimicrobial resistance. In short form, I will term it as AMR. And then thirdly, to look at the genetic diversity of the selected Seroa Brancaster in my study. So for the research method, so for my study, I used a total of 100 Salmonella isolates archived in our institute. 68 is from the culture swab, meaning from live chicken from the poultry farm, and then 32 is from the whole chicken or the parts collected from the abattoir. And then the sample collection is actually done from our staff under this routine Salmonella surveillance program between 2017 until 2022. And the isolation, we follow the standard method. And then for the serotyping, it will be all done in our institute. And then this is the pure Salmonella Brancaster culture, which I will use for my further testing. So from the bacteria, I extract the DNA just using the boiling method. So the supernatant will be used as the template for the PCR test. So in this slide, for objective 1, to study the virulence, so I will detect the virulence gene using the PCR method in multiplex. So it will detect 8 virulence gene in 2 runs. And then for objective 2, to look at the AMR profile, I use the disk diffusion method that I test against 9 antibiotics. And all these antibiotics is the one that are frequently used in farm and also monitored under the department. And then I further test those isolate that show the penta-resistant pattern. This one, the ACSSUT. This one, the normal terms is penta-resistant. They're resistant to 5 types of antibiotics. That is ampicillin, chloramphenicol, streptomycin, sulfonamide, and tetracycline. So this penta-resistant strain, I will continue to further test to detect the 12 resistant genes corresponding to the resistant pattern. And then for the third objective on the genetic diversity, I use the multi-local sequence typing method, where we will amplify the 7 housekeeping gene, and from the housekeeping gene, we will look at the allelic profile. This one is the standard method for sequence typing. And then at the end, the further data for the nucleotide DNA, we will do the dendrogram using the Mega 11 software. So for my findings, in objective 1, the Salmonella Brancaster were found to carry multiple virulence genes, and all these virulence genes were present. And this SPVB gene is just present in one isolate. And all the virulence gene, they actually encodes for the secretion system that will play an important role in the pathogenesis of the Salmonella. And in my finding also, I found that 26% of the Salmonella Brancaster, they carry this cytolithal distending toxin B gene. And this finding is interesting because previously this gene is only found in the human restricted Salmonella typhi strain. And then now we are detecting it, this one is from the chicken origin. And this is of major concern because it could cause invasive cases of human bloodstream infection. objective to the AMR profile, the finding is that 85% of my isolates were categorized as multi-drug resistant, meaning they resistant to more than 3 groups of antibiotics. And the highest resistance is to ampicillin, up to 91%. And then 1/3 of the isolate exhibit the penta-resistant pattern. And 7 out of resistant genes were detected. The highest one also, this one is towards the ampicillin. So for the genetic diversity, Salmonella Brancaster in the present study indicate a single sequence type, ST2133. And this is in agreement with other publications from the Asian region. We are having the same lineage. However, in West Africa, there are Salmonella Brancaster having a different sequence type. They have the ST3704. So their lineage is different from ours one. And the further MLC data here divided my 10 isolate into 9 cluster. The figure here, the 86%, this one indicates the confidence level, meaning that if more than 70%, it's strongly supported that these 2 isolates belongs to the same cluster. So from my 10 isolates compared to the reference strain from Singapore, I can get 9 clusters. So this meaning that our isolate itself is very diverse and heterogeneous, and they were never 100% identical. And all this variation is caused by the nucleotide substitution, insertion and deletion, caused by maybe mutation, the acquisition or loss of the genetic material over time. So overall, for my findings from my research project is that number 1, Salmonella Brancaster isolated from chicken and raw chicken meat has the potential to be virulent, since it possesses all the virulence gene tested, similar to other major medical relevant human and veterinary seroas. And secondly, the Salmonella Brancaster, they were found to exhibit a high level of antimicrobial resistance, and they carry multiple resistant genes. And for the third objective, the Seroa Brancaster, they were identified as sequence ST2133, and they were highly diverse. So for the significance of my study, Salmonella Brancaster is present at the farm level, meaning at all the poultry farm, and then we found it in the chicken meat also, meaning at the logic and process and everything. So it suggests that it may be in our food chain production, and it might move up to the consumer level also. And the emergence, persistence, and dissemination of this multi-drug resistant Salmonella Brancaster, which carry all the virulence gene, they actually raises the public health concern. And one of the concerns is the high level of ciprofloxacin resistance, 9%. Because previous publication, they only reported from 0 to 3%. For our isolate, it's up to 9%, and there is a very high intermediate resistance, up to 82% also. So this suggests the possible emerging of the ciprofloxacin resistant Salmonella Brancaster. And this one I highlight because ciprofloxacin is the important antibiotic for human salmonellosis treatment. This characterization study to study the bacteria is very important so that we can know the pathogen propensity for infection, and also to generate an up-to-date epidemiological data for our diagnostic, prevention, and control strategies. So from my research finding, we found that the Salmonella control management in farm needs to be periodically reviewed and updated to improve by including the emerging Seroa and the dominating Seroa as a possible pathogen of public health concern in our country. So we should look at the basic point. They should emphasize on the biosecurity and sanitation practice in the farm. And also our salmonellosis control management is written in our DVS policy, the Protocol Veterinaria Malaysia, this one is the latest edition 2021, which draws the outline the policy, meaning all the Salmonella positive cases is categorized as notifiable disease, penyakit wajib lapor. From the farm to or the testing lab, they must report to the state DBS, and then from state they will channel the information to Putrajaya, our HQ. And then there is another document that is implementing is the MyGap, Malaysian Good Animal Practice Guideline. So in this guideline also, it is important to reach the high standard in farming. So in this guideline, they will cover the application, certification, and audition process. And the inspection and auditing will goes on until they reach the standard for the MyGap certification. And then for this slide, the research implication is on the AMR. Just now the antimicrobial resistant issue. So actually AMR is a global issue, and actually it's a One Health issue also that involve human, animal, and our environment. So in farm animals, actually small dosage of antibiotic is a common practice. They always administer a small dosage to the, especially in the commercial poultry farm, so that they can reduce the stress and to prevent the disease in animal. So in this Malaysian Action Plan on antimicrobial resistance, which started in 2017 and launched in 2018, which include the collaborative work from a few ministry and agencies, include the Ministry of Health, Ministry of Agriculture and Food Security, Ministry of Higher Education, Ministry of Education also, and then they work together with the private sector. Let's say for the hospital, the community pharmacies, and then all the NGO, they were working together to draw out this National Action Plan, and the latest edition is here. Starts from 2022, so it's an ongoing process that draws out the 4 key priority area. The first one is the public awareness and education, to improve the awareness and understanding of AMR through effective communication, education, and training. And this one they will provide it in school as well for our school children. And the second one is the surveillance and research, to strengthen the knowledge and evidence base through surveillance and research. So my research is under this key priority. And for the third one is the infection prevention and control, is to reduce the incidence of infection through effective sanitation, hygiene, and infection prevention measure. And this one also is an ongoing effort. And the final the fourth point is the appropriate use of antimicrobials, to optimize the use of antimicrobial in across all relevant sectors. And also for this key priority number 4, actually they are those that involved in this National Action Plan. Actually they are taking the proactive measures to ban and phase out some of the important antibiotics that used in animal production. They try to phase out the usage for farm animal, such as colistin, erythromycin, and all. So it is an ongoing and they are at a progressive stage. And then with this, before I end, I would like to express my gratitude to my supervisor, Dr. Norinda and Prof. Sunita from UPM, and then also Puan Rosliza and my pengarah, Dr. Faiza from the DBS, and also thanks to JPA for their support of my study. Thank you.
Yes, Rashid. Come forward, sir.
Good morning. My name is Muthu from Kementerian Perpaduan. Actually, I congratulate for taking this kind of very useful research. It's really wonderful thing for us. So actually, I'm not interested on living chicken which not taking bath with bar soap and shampoo daily like us. Okay, so I'm much interested on raw meat. Okay, so kindly explain what about the possibility of salmonella in raw meat, which may be found inside not cooked well. Okay, sometimes we open the chicken, we can find blood stain. The blood is still coming because maybe they put so long inside the freezer. So when they cook, it's not cooked well. It's really a common thing happening nowadays. Wherever you go to the restaurant, you go for any ceremonies, you usually can see that. Okay, you cannot avoid. Even you're eating chicken rice or curry chicken, also you can find this kind of thing. So can you explain about what is the possibility existence of salmonella in that, and how to overcome the salmonella? And kindly explain about the Brancaster, how strong Brancaster compared to other salmonella stereotypes. Thank you.
Thank you. Thank you for the question. For the first question is about the salmonella in chicken meat itself. So why the salmonella is there? Because they carry along from the farm. From the farm, they must go to the processing facility, the logi, and then actually the way they process the meat, the whole processing is the hygienic is quite a concern because from the whole farm, they must go the logi, and then they are using the same tank of water to rinse everything until the storage. So what I can say from our point as a consumer, that's why the public awareness is the first thing. So when we notice there is something not normal with the chicken that we eat, so better not to take. And then also we must choose our reliable source. And then we ourselves also must know how to store it properly. Yeah, most of the food poisoning case, actually when they do the epidemiological trace back, right, it's not the farm that have the problem, it's the human error, how they handle, how they place it exposed under the high temperature. That's where the bacteria, if undercooked, the bacteria will start to grow with the temperature. Okay, so for the second question is about the Brancaster, is it? Actually, just now the enteritis and the typhimurium, that one is the most common and the one that we don't want in our product because that one have the zoonotic potential, meaning can infect human to cause all the diarrhea and vomiting. Very serious, that one. So in our context, because I'm from the lab, so every salmonella positive isolate that isolated from the regional lab all will end up in VRI. So I can see the data and I see the trend. That is where I think in 2018 we noticed the high rise suddenly. Why so many Brancaster? So that's in our mind because we are working in the bacteriology lab and we are also a team of researchers inside there. So that's where I start to ask some information from KKM because newspaper everything they doesn't report on the Sirowa. Even I think the last month the food poisoning case they didn't review what Sirowa also. So that one also I'm wondering what type of Sirowa is it? Because when I start my study in 2019 when I write the proposal, at that time not much food poisoning case in our country reported. Very little. So meaning news report and then publication journal also cannot find salmonella outbreak. If have also maybe 20 years ago, they were talking about typhimurium and enteritis. Nothing on Brancaster. That's why I'm surprised where at that time the journal publication is 1952 for the first one. So from our research finding also, the Sirua branchiaster have the potential to become pathogenic because it contains all the virulence genes, just that maybe it doesn't express well like other Sirua like Enteritis and Typhimurium. So that's why in human, we don't see any outbreak yet. So with thorough cooking, everything actually can kill the bacteria.
Can you explain how the thorough cooking? What is thorough?
Masak, no red and all the liquids.
Untuk masyarakat umum, public, I think the public is good to know when they buy chicken. Is it menolak free? One. Kemudian menolak bakteria ni, is it temperature sensitive? Maknanya waktu dia masak, dia mati. And then how many hours lepas tu dia ada balik? Sakit sekolah yang mati makan telur tu, I think dia kedai tu bagi telur yang after 10 hours. After 10 hours datang balik bakteria yang mati tu kan.
Yeah.
So dan
That's why
chicken
it's a storage problem.
ada tak? Bila dia beli itu kan dia tahu dalam beli tu ada salmonella, dia masak, and then dia mati within 3 hours or so. Apa pengalaman dia?
To trace food poisoning, I don't have the pengalaman because I'm a lab person. So for investigation, I just listen from their report. So the whole process of interview, right? You see the interview, how long you store this one is very interesting. So we can see that the way they handle the food is really not parts at all. Meaning they expose. Maybe the thing is already cooked, and then salmonella dia mati at 60 degrees. That one theoretically, but actually we don't know, right?
Saya rasa tadi dia daftar bila masak after 3 hours tu mesti makan. Mungkin ada kan? Dia daftar.
Handler is also an issue. Actually, this Salmonella branchiaster is multiple hosts. Other animals also got. Because some Sirua, they are only restricted to birds, but this one multi-host. Just now that Enteritis also and Typhimurium also multi-host.
So what would you suggest for I would say policy, we don't have many policies in place. What are the actions that can be taken to curb this? Because we don't actually have the mechanism to monitor what goes into our body. We don't even know what's in it. So anything that you can suggest that you guys can push forward for us to wear off? Orang campaign dah terlalu banyak buat something that we can flag for this is very dangerous that we can recommend. And everybody here is very lucky to know the recommendation so that we tak makan salmonella. Yes, right.
If recommendation if in our department, we are taking the effort to revise back the PVM, the Protocol Veterina Malaysia, whether to include the currently dominating Sirua in the control prevention measures. Meaning, if farm detected got this one, so if it's stated there which Sirua highlight red color one, they must report and take immediate action. That one is at the farm level. Consumer is not under our regulation. So that's why we always say it's always about public awareness. That's why when we do this research, we try to go for conference. So we try to walk around.
Any more questions from the floor? Yes.
Hi, since you are a lab person, so what do you suggest as normal lab, not lab person, to choose which kedai yang we should eat kita nak makan kat luar? So based on your, sebab you dah tahu kan yang salmonella is like this, so you based on your observation, you must be macam very particular on eating outside kan, macam okay this kedai, okay this is not for me, sebab mungkin dia punya hygienic dan sebagainya. So how do you suggest us yang kita orang tak berapa nak family on this kind of bacteria untuk memilih kedai yang better untuk avoid this bacteria? Thank you.
I think the basic one everyone also knows a little bit. Just that additional from that, from what my observation those kedai that you can see all the birds coming in one, avoid that one, because wild birds also is a carrier for salmonella. So, thank you.
Ada soalan lagi? Yes, please.
Selamat tengah hari. Nama saya Rahim. Saya inspektor kesihatan daripada Pejabat Kesihatan Daerah Kelang. Jadi sebenarnya sangat berkaitan lah. Macam saya pernah bertugas di food safety unit lah untuk daerah Kelang selama 3 tahun. Cuma soalan saya macam salmonella ni, macam doktor kata data tu daripada news saja. Saya tak buat doktor pernah tak? Puan pernah tak? Ambil data daripada, ada satu unit dalam Kementerian Kesihatan ni, dia panggil bahagian kualiti dan keselamatan makanan. Macam saya pun terlibat ambil sampel makanan ni. Sampel salmonella ni memang boleh kata for 3 years, every year mesti ada. Dan semua daerah di Malaysia sebenarnya akan ambil. So data kalau nak salmonella punya taburan data satu Malaysia ni, puan boleh, saya sarankan kalau puan boleh dapat data tu daripada bahagian keselamatan dan kualiti makanan di bawah Kementerian Kesihatan. Jadi mungkin boleh more data, boleh lagi itulah. Itu cadangan saya. Lepas tu itu sajalah. Saya bukan nak tanya kan, bagi cadangan.
Thank you.
Itu saja. Terima kasih.
Thank you. Now we know mana-mana kedai ada burung merpati, burung liar jangan masuk. Nanti kena salmonella dengan ada-ada. Thank you Puan, thank you so much.